Hello everyone and welcome to this video on the biochemistry of the Huntington disease. The Huntington disease is a disease that is caused by an autosomal dominant allele that is usually characterized by uncontrolled movement due to the death of brain cells. It is autosomal. That is, it is carried on the non-sex, the mutated allele is carried on the non-sex chromosome or on the autosomes. That means, in other words, that gender does not have an effect on the distribution of this mutated allele. So either male or female have an equal chance of having the disease. It is dominant. That means that a single copy of the mutated allele is enough to cause the expression of this disease. Let's take this example below, the cross between an individual that is heterozygously sick for the Huntington disease and this other individual who is normal. Note here that the Huntington allele is represented with a capital H because it is a dominant or an autosomal dominant allele. So a single mutated copy in this individual here causes the individual to have the Huntington disease. So when we do that cross, we'll notice that the phenotypic ratio of sick individual to normal is 1:1. Okay, so if we look at the resulting offspring, the F1 generation, we'll notice that these two individuals are sick because of the presence of one allele of the Huntington disease, and these other two individuals are normal. So the phenotypic ratio of sick to normal is 1:1, while the genotypic ratio of this individual with capital H, small h, that is heterozygously sick for the Huntington disease, to small h, that is a homozygous normal individual, is also 1:1. Okay, because if we look at these two individuals here, they have the genotype capital H, small h, so they are heterozygously sick, while these other two individuals have the genotype small h, small h. So what actually causes the Huntington disease is DNA slippage, which is due to the expansion of the nucleotide triplet CAG above a certain threshold. Okay, so this means, in other words, that this triplet usually occurs normally on the genome, but when it expands above a certain threshold, it now leads to the Huntington disease. So individuals with triplets, that CAG triplet, let me put it, the CAG triplet less than or equal to 26 do not usually develop the Huntington disease. Individuals with repeats between 27 to 35 do not also develop the Huntington disease, but this repeat can expand in their offsprings to give rise to the Huntington disease. Okay, individuals with repeats between 36 to 39 have mixed expression of the disease. Some develop the disease, the Huntington disease, while some do not. Okay, while individuals with repeats greater than or equal to 40 usually develop the Huntington disease. So we see that above a certain threshold, there is the expression of the Huntington disease when these triplets go above a certain threshold. Okay, and so also in the Huntington disease, we have the phenomenon of genetic anticipation. That is, there is an increase in the severity of the disease as we move from one generation to the other. That phenomenon is known as genetic anticipation. Let's look at this pedigree here on the right. Although this example is not for Huntington disease, but it's a similar situation for Huntington disease. This is myotonic dystrophy. Okay, this pedigree is for myotonic dystrophy. That we want to take it to show the phenomenon of genetic anticipation. If we look at this first generation, this individual here, the person has myotonic dystrophy. We see that it was diagnosed at 55 years, and the signs and symptoms, we have cataracts and 120 repeat. When we move to the second generation, we see that this man here, this individual here, this man here, it was diagnosed even earlier, that is at 50 years. The signs and symptoms involve bilateral cataracts, facial weakness, myotonia, cardiac problems, and the number of repeats have increased, that is 700 repeats. When we look at this female here, also of the second generation, we see that it was diagnosed also even earlier at 48 years. The signs and symptoms involve grip myotonia, facial weakness, and we have the number of repeats have also increased, 500 repeat. Then when we move to the third generation, this individual here, we see that the disease was diagnosed even earlier than those of the second generation, was diagnosed at birth. Okay, signs and symptoms involve facial weakness, severe neonatal hypotonia, and the number of repeats have gone even higher, that is 1,200 repeat. Okay, so we see the concept or the phenomenon of genetic anticipation. That is, the severity, the disease becomes more severe as we move from one generation to the other. Now, in the causes or the cause of the Huntington disease, we talked about DNA slippage. So what is DNA slippage? Let's try to explain it looking at this figure below. Okay, so let's consider that this is a double strand DNA. Okay, and for this double strand DNA to undergo DNA replication, that DNA double strand has to be unwound by this enzyme helicase. This helicase, which we have represented here by this green spherical ball. Okay, so helicase has to unwind it. So what does helicase do? It comes in here and then unwinds the DNA double strand, okay, and pulls it up, forming a loop. Okay, so you see that the DNA helicase has pulled this triplet upward, these two triplets upward, forming a loop. Okay, due to the fact that the triplet is a repeat, it is repeating itself. So when it pulls it upward, forming this loop, we will see that this fourth triplet will come and pair up with the second triplet of this other strand below here. Okay, and when this happens, the other enzyme, DNA polymerase, which we have represented here with this yellow ball, DNA polymerase will come and then copy this upper strand, goes up, copy, okay, comes back and will continue to copy in that direction. Okay, likewise, it will also copy this other strand below. Okay, when this happens, it gives rise to DNA strand, two DNA double strands with unequal length. We see that this other strand up here has two additional triplets. Okay, while this other double strand down here has the same length as the original DNA double strand that was unwound from the beginning. Okay, this is due to DNA slippage, and this occurs because there is a repeat. The triplets are repeating, okay, such that when RNA or helicase pulls this triplet upward, okay, this triplet here can come and pair up with this second one here, and DNA polymerase will come and copy in that direction, therefore giving two DNA double strands with unequal length. Okay, and then when this occurs, it keeps repeating itself and then keep on adding this triplet. So this process will keep on repeating itself with each cycle of DNA replication, adding this triplet, keep adding this triplet until it goes above the threshold of 40 triplet repeat. Okay, so and when that happens, it will result in the Huntington disease. So that is it for the biochemistry of the Huntington disease. I hope you were able to learn something from that, and we wish that you should also subscribe to the channel so that contents like this, when it is uploaded from my channel, you can be notified. Okay, so thanks for following the video.